So welcome to the KCA lecture series. We're pleased today to have Nolan Wages from VCU Massey Comprehensive Cancer Center to talk about trial designs for early phase clinical trials. Many of you are in this space or lab translational, thinking about application to clinical trials. And having been around the clinical research space for a long time, we tend to sort of do things the same. Some of that's good, some of that's not good. So I'm excited to hear about some novel designs to test these concepts. So Nolan, we appreciate you joining. I'm going to turn it over to you. We may interrupt with questions, and we'll certainly leave some time at the end for that, but we appreciate your time and expertise.
Sure. Thank you for having me. Thanks, everyone, for being here. Yeah, I actually can't see anyone in the presentation mode, so if you have a question, please just feel free to interrupt me. This can be conversational and not me lecturing the whole time. So, yeah. Yes, again, thank you for having me. And I'm going to talk about novel clinical trial designs for early phase trials. And yeah, it's somewhat of an ambitious talk in terms of number of slides. So we'll go ahead and get started. And this is the traditional dose finding. We're talking about dose finding trials in this talk when we talk about early phase clinical trials. And this is sort of the traditional setting. Some of you may know this, but the primary objective usually in these trials is to identify the highest dose with an acceptable DLT rate. Dose limiting toxicity is our primary endpoint, usually in an early cycle of treatment. That is DLT is defined by protocol specific adverse events, and we usually collapse this into a binary endpoint. Did the patient have DLT, yes or no? And our goal is to find the highest dose with an acceptable DLT rate, which we term the maximum tolerated dose. So the figure here gives sort of a general schematic for how these trials work. We have a small cohort of patients that we assign to one of the dose levels that we are studying. In that small cohort of patients, we observe the number of DLTs, and then we recommend a dose level for the next cohort based on some design algorithm that we have, and we'll talk about several today. And then we repeat this outcome adaptive process for usually a sample size of N participants, which is usually small. We're usually talking about patient numbers in the upper teens or 20s for these types of trials. So this is the general idea or the general setting we're going to be working within, and then we're going to build on this a little bit throughout the talk into more complex settings. But this has historically been the dose finding setting in which a lot of designs have been developed. So the primary assumption that's driving the design of these trials is that as the dose of the drug increases, so does the probability of toxicity or DLT. And that's represented by the red curve here. So on the x-axis, we have dose or drug exposure. On the y-axis, we have outcome probability. And as the dose increases, so does the probability of toxicity or DLT. But also driving an assumption driving the design of these trials is that as dose increases, the probability of efficacy also increases. We're going to relax that assumption later in the talk and talk about some designs that are able to be used when that assumption does not hold true. But the idea that we're finding an MTD, we're pushing the dose as high as we can up until some acceptable DLT rate, against some acceptable DLT rate, is really driven by the assumption that as we push the dose higher, we don't want to go beyond that safety threshold. But that dose has also given us the most promising outlook for efficacy, which is one of the reasons efficacy has historically not been used in this phase of development as an endpoint to drive dose allocation, but it's been based primarily on safety. So this assumption, again, is in the classic sense, is driving the design of dose finding trials and really is geared towards cytotoxic chemotherapy. And again, we're going to relax this assumption later in the talk. So generally, phase 1 trial designs or dose finding designs can be classified into 3 different buckets. The most classic is a rule-based algorithm. Some of you may have heard of the 3+3 algorithm. We're going to talk about that briefly in a moment. That's the most commonly used dose finding design. in terms of chronologically how these designs were, when these designs were developed, next in the chronological order was the class on the far right, which is model-based continual reassessment method, or CRM, that was proposed in the early 90s. And then a newer class of designs in the last decade or so is model-assisted designs. And one of these is the Bayesian optimal interval method, or BOIN. You might have heard of that one. And before it was the MTPI, modified toxicity probability interval method. So we're going to talk about each of these classes of designs, and then we're going to focus our attention on model-assisted and model-based for extensions to more complex problems that we might encounter in this space. So what's used typically in oncology trials? This is a very recent report on which methods were used in trials published from 2020 to 2022. You can see that these authors evaluated 218 dose escalation studies, and the 3+3 was used in 161 of them, so that's 74%. The CRM, or some modified version of that, was used in only 9 trials, that's 4%, and the BOIN method was used in 3% of the trials, or 7 trials. So you can see that still the 3+3 to this day is the overwhelming choice for designing these trials in oncology, and that percentage has probably gone down a little bit over some previous published studies. I think there was one done from 2006 to 2011 or so, and that percentage was close to 90 or in the low 90s. So it's coming down a little bit with an uptake of some other methods, some more novel methods, but still it's highly prevalent and used quite a bit, the 3+3. So here's how the 3+3 works. On the right, you'll see a schema for how this works. We're going to enroll 3 patients on our starting dose level, or on wherever dose level we're currently accruing patients, and then make one of 3 decisions based on the number of DLTs we see in that cohort of 3. If we see 2 out of 3 at least, we're going to stop the trial, and some lower dose, the dose immediately lower than that, is deemed the MTD. If this happens at the lowest dose, the trial is stopped for safety, and no other doses are explored. If 1 out of 3 DLTs are observed, we enroll 3 more patients at that dose level. And if 0 out of 3 DLTs are observed, we escalate to the next dose level. In the second cohort of 3, if we were to see 1 out of 3, if total we see 1 out of 6, we escalate the dose. If total we see at least 2 out of 6, we stop the trial and declare the lower dose, the dose immediately lower, as the MTD. So one defining characteristic of this algorithm is that only 6 participants will be treated at the selected MTD. And operating characteristics wise, the statistical properties of this is that has a low probability of selecting the true MTD. It's not very accurate, and many participants are treated at subtherapeutic lower dose levels. So again, remember, the assumption that's driving the design is really trying to balance a trade-off, right? We don't want to push the dose too high because we don't want patients treated at overly toxic dose levels. We don't want to treat the patients too low because there's a low probability of benefit. So we're trying to strike this balance between not exceeding a safety threshold and getting some benefit out of the drug. So this is a common approach to this type of problem with the left side of this screen, the left side of the blue line, illustrating that we perhaps a common approach is to conduct a 3+3 design on these dose levels. We're only going to have 6 participants on the selected MTD, as we discussed in the previous slide. And then we're going to go to part 2, which is dose expansion, and we're going to use the dose we identified from part 1 to put an additional 15 participants on that dose for a total of 21 selected on the selected MTD. This is a common approach that I see in a lot of protocols that I review, with dose expansion happening only at this one dose level that we've identified from part 1. This schema actually came from a real protocol that I reviewed. This is quite a common approach in this space to sort of get further data on the dose in the dose expansion after we've decided on it with a small number of patients in the dose exploration or dose escalation. The issues with this approach is that expansion is conducted only at that selected MTD historically. So that's based on a small sample size. As we've seen, that's only 6 participants are going to be treated at one of the dose levels before we move that dose level to the expansion cohort. But it begs the question, what if multiple DLTs start to be observed early in the expansion phase? If we have 1 out of 6 DLTs on the MTD and then move it to expansion phase, but then the next 2 patients have a DLT in the expansion phase, then we have 3 out of 8. And there's not really good mechanisms for adjusting the dose within that expansion cohort. It usually requires a protocol amendment. But on the other end, what if we observe no DLTs in the expansion cohort? Does it suggest that we found too low of a dose? So if we had 1 out of 6 and then we put 15 more on and no DLTs are observed, we've only observed 1 DLT out of 21 patients, suggesting that we might be at the dose that's too low. So in these cases, the protocol rarely includes transparent rules for formal safety monitoring in that expansion phase, adjusting the MTD, and these things require protocol amendments, which might cost time and resources and be inefficient. So it's not always great to fix the expansion dose at the MTD, and that may not always be optimal. So we're going to talk about these designs, BOIN and CRM, in detail in just a minute, but I just want to point out that these web applications for these designs, BOIN and CRM, have mechanisms that you can specify to stop the trial if the number of patients assigned to one single dose reaches some value and you give it the value. So for example, in the example that we saw with the expansion phase, we had 15 in the expansion phase, 6 on a dose in the escalation phase. We're going to have a total of 21. Well, these designs just allow you to sort of keep the dose escalation open. You move doses if you need to, right? If you start seeing DLTs, you move the dose down. If you start seeing non-DLTs, a lot of those, you move the dose up. And you just do this until you get 21 patients on a dose level. Again, this can be specified to be any value. I put this as an example, but this allows you to just sort of keep the dose algorithm open and move the dose as needed until you reach a pre-specified number on a dose level. So one of the advantages of these designs, among others, is that you can specify, or you will in the end have more than 6 patients treated at your MTD, and you can have more precise estimates of what the DLT rates are at that dose level. DLT rate is at that dose level, and probably have more precision that and more confidence that that is your MTD. So these are other design characteristics that I wanted to highlight among the different classes of designs: algorithm-based, model-assisted, model-based. In the red box, it's highlighting that with algorithm-based designs and model-assisted designs, the de-escalation and the dose de-escalation and escalation rules can be predetermined and included in the protocol. So like the 3+3, I know if this happens out of 3 patients, I'm escalating the dose or I'm de-escalating the dose. Model-assisted designs, as we'll see, also have that property. All of the rules are transparent and are able to be put in a protocol. This is not true for model-based. In short, the reason for that is that you're using all of the data at every dose level throughout the trial to make decisions. And in model-assisted and algorithm-based, you're just using the data at the current dose level. So that creates a little more complexity with the model-based design, and you're not able to include all of those rules in the protocol. The number of patients, we just touched on this, the number of patients treated at the MTD can be greater than 6 with model-assisted and model-based designs, but not with 3+3. And identifying the MTD accurately, again, that's our goal, right? If we want to find the MTD, we want to do that with some accuracy, and model-assisted and model-based do that a lot better than an algorithm-based design. Here's some implementation barriers to implementing novel designs. My reference here got blocked by the words, so this came from a British Journal of Cancer paper by Love and co-authors in 2017. The clinical investigator preferring the 3+3 is the most common barrier. The clinical investigators' lack of knowledge of novel designs and how they work, that's what that means there, is another barrier. So I'm hoping that today's talk alleviates one or both of these things, that we can get a little more knowledge about the novel designs and perhaps prefer them to the 3+3. So let's start with CRM. We'll go through an illustration of this. In the CRM, we're not going to get too statistical here, but in the CRM, a simple dose toxicity or statistical model is assumed that's going to model the relationship between dose and toxicity, relationship between dose and DLT rates. And we specify a target DLT rate that defines our MTD that typically ranges in values from 20% to a third. Let's say it's 25%. And the schematic is fairly simple. We assign the patient to a dose level with an estimated DLT probability closest to the target, and we have a starting point for that when we're starting the trial that we'll look at in just a minute. We observe whether a patient or a cohort of patients has DLT or not, and then we update the estimates of the DLT probabilities at every dose level, and we repeat this process. Once we get these estimates, we assign the next cohort to the one with an estimated DLT probability closest to my target of 25%. So we're using the statistical model and the data at every dose level to make a recommendation for each incoming cohort of patients. And the MTD is the current dose recommendation after N participants have been accrued, or we have some stopping rule that's predefined in the protocol. So here's an example of a study that was published in, I think, in 2019. You can see the reference at the bottom there. Again, it's hidden by the words. This is a phase 1 study of pevonedistat in combination with docetaxel in patients with advanced solid tumors. The docetaxel dose is fixed, so we're not escalating that. It's really not of interest here. Really, what matters is the escalating doses of pevonedistat. 4 dose levels: 15, 25, 37, 50. We have a target DLT rate of 20%, defining the MTD, maximum sample size of 20 patients. So this example is loosely based on the paper there in 2019. So this is just going to give a brief illustration of how CRM works. We're going to start with what's called a skeleton for the probability of DLT at each dose. This is sort of an initial guess of the probability of DLT at each dose level. I could give a talk on how these are chosen, but in the interest of time, I'm not going to go too much into that. There's people that have done a lot of work on how to sort of get a CRM trial going, and there's systematic approaches for choosing these values, and software that does this. So it's really an automated process. But in the long and short of it is these initial guesses here: 0.2, 0.31, 0.42, 0.53. The method's fairly robust to this choice, so long as these values are somewhat adequately spaced from one another. Let's think about a couple of extreme cases. If we space these values really close to each other, like 0.2, 0.21, 0.22, 0.23, our method's not going to be able to distinguish between the doses. Basically, we're saying before this trial starts, we have every dose is about the same. The data, we're not going to have enough data to sort of separate those dose levels out. On the other end, if we say use very extreme values that are steep, for example, 0.01, 0.2, 0.8, 0.9, we're heavily biasing that dose level 2 with the 0.2 is the MTD, and we don't have the amount of data that's going to be able to overcome that heavy prior assumption. So basically, we just need to sort of adequately space these values, and we can get started. What the statistics behind this is, we're going to estimate this parameter A as we accumulate data. I'm not going to give mathematical details on that in this setting, but we're just going to see how it works in the presence of a small amount of data here. So let's say we start at dose level 1. We're going to put one patient on dose level 1, and then we're going to update A and see what happens to this curve that we started with here, which is 0.2, 0.31, 0.42, 0.53. So let's say the first patient gets dose level 1, does not have a DLT. So now this is my data: 0 out of 1 at dose level 1, zeros at all the other dose levels. I do my statistics, and I get this A. I get an estimate for this A to be 1.31. I update. I take my skeleton. I put it to that power. Here's my new estimates: 0.12, 0.22, 0.33, 0.44. What happened to the curve? My previous curve was the green one. My updated curve is the red one. All we need to take away from this is that this green curve, once I had a non-DLT, it shifted down, and look what the next dose is, the one that's closest to my target line, which is dose level 2. So this is going to escalate to dose level 2. I heard a question. Is there a question? Or maybe not. Okay.
I do have a question.
Sure.
This is Rainier from University of Wisconsin. So isn't that like a little bit against article, like I don't think logic, that if you don't have a DLT now, the curve shifts down instead of up?
The curve should shift down because we have right. If we had all non-DLTs, right, it would indicate that the toxicity probabilities are very low. So in the presence of non-DLTs, we should shift the curve down. In the presence of DLTs, the curve should go up. So if we had a lot of DLTs, right, the curve would be up here.
Okay. Oh, I see.
Right.
Thanks.
Yeah. So you see that this one DLT shifted the curve down. Now my next dose level is dose level 2. I'm going to put the next patient there. Just one more illustration here. You see a non-DLT at dose level 2 shifts the curve down into where we get dose level 3 as the next recommendation. In the presence of DLTs, again, the curve's going to go up. Basically, this is just this estimation of this A parameter is just shifting this curve up and down and moving it or trying to see which estimate is closest to my dotted line or my target rate. It's just a mechanism for moving the curve up and down. Somebody once, when I gave this talk, asked me, or they described this in their words as the CRM is sort of like playing Battleship, right? We're trying to hit our target. We're moving up and down. We'll see if we can, which dose is going to get closest to the target line here. I liked that analogy. So this is what it would look like at the end of the study, right? We would have open circles be non-DLTs, dark circles be DLTs. In the presence of black circles, right, we move down in general, right? And in presence of open circles, white circles, we move up in general. But eventually the method sort of settles down at our MTD estimate, and we are able to accumulate 11 patients out of 20 at dose level 3, and we observe 2 DLTs there. And our final estimate is 0.18 for the DLT probability at that dose level. That's closest to my 0.2, which is my target. So dose level 3 would be selected as the MTD. That's just a little bit about how the CRM works. So CRM, as I mentioned, has been thoroughly studied since 1990, and it has recommended design specifications. Again, a lot of people have done work on how to sort of automate this process, how to choose that skeleton. There's some work there. Which statistical model to use. You don't have to use that skeleton raised to a power of A. You can use other models like logistic regression model. How to choose a prior distribution if you're using a Bayesian approach here. And all these recommendations make CRM more easy to use than it was in the 90s. And these recommendations yield appropriate dose assignments and provide a good chance of finding the MTD at the end of the study. And we have a CRM app that we developed that can be used to design these studies and make it easy to design these studies and implement. So you have a tab for simulation and a tab for implementation here to be able to make this process more efficient. So let's talk about model-assisted designs. The motivation for these was to introduce a platform that's easy to implement in a transparent way like the 3+3, but yield comparable performance to more complicated model-based designs such as the CRM. And this type of design can handle both single agent trials and drug combination trials, which we'll talk about in just a minute. So we're going to talk about one specific model-assisted design, which is called the Bayesian Optimal Interval Method, or BOIN. Let's suppose we have a phase 1 trial of 4 dose levels like we did in the CRM and a target DLT rate of 25%. This flow chart simply explains how this works. So we're going to start at some pre-specified starting dose and treat a cohort of 3 patients at that dose. It could be another cohort size, but this example has 3 patients as the cohort size. We evaluate whether a stopping rule has been met. If not, we go down here, and here's where the red circles are. This is the basis of the design. We're going to compute the DLT rate at the current dose level. So how many DLTs do we have at that dose, and how many patients have we put at that dose level? And if that rate is below, in this case, related to a 25% target rate, if it's below 20%, it's indicating that this dose is safe, so we should escalate the dose. If it's greater than 30%, it's indicating that this dose is unsafe and we should de-escalate the dose. And if it's between 20% and 25%, it indicates that we're in a range that we want to be in, around the target rate, so we should stay there and retain that current dose. So it's basically a sample proportion. How many DLTs do we have over the number of patients that have been put at that dose, and then making one of 3 decisions based on where that rate falls relative to these 2 values. And again, the mathematics behind this is how these values are derived. But again, this doesn't have to be thought of in actually conducting one of these trials. This is sort of an automated process for how this interval is chosen. So let's do a trial in which we have a maximum sample size of 21. So this is how the decision tables look. On the x-axis, we have the number of patients treated at the current dose. And on the y-axis, we have the number of patients with a DLT. And D is de-escalate, S is stay or retain the current dose, E is escalate, and DE is de-escalate and eliminate that dose. For example, here's 3 patients at that dose level, 3 DLTs. If we had 3 out of 3, we would de-escalate the dose and never try that dose again. So this is how this decision table breaks down. So let's look at a simple example here. So again, open circles are non-DLTs, colored circles are DLTs. Let's say we put the first cohort of patients, which is 3 participants, on the first dose level, and we have no DLTs. We go to our x-axis of 3 patients, our y-axis of 0 DLTs. That tells me to escalate. So I'm going to put the next patient and cohort of patients on dose level 2. If I do that and there's no DLTs, I'm going to use the same decision process. 3 patients, 0 DLTs, escalate the dose. I'm going to move up to dose level 3. Same decision. 3 patients, 0 DLTs, move up the dose. Here I have 1 out of 3 on dose level 4, targeting 25%. If I observe a 33% rate, that's above the upper bound of that interval we looked at on the flow chart. So I'm going to de-escalate the dose and go back to dose level 3. So you can see I have 3 patients here, 1 DLT tells me to de-escalate. Once I do that, I'm going to put a second cohort of 3 patients on dose level 3. And in total now I have 6 patients at that dose level. Again, we factor in all the patients that have been put there. And we have 2 DLTs. So I go to 6 patients here on the x-axis, 2 patients have DLT. I'm going to de-escalate the dose. I go back to cohort 6. I'm going to use all 6 of these patients and I have 0 DLTs. I'm going to escalate the dose. And I go back and I put my final cohort on dose level 3, and I have 0 in that cohort, but I have a total of 2/9. And that's the end of the trial. So that's recommending that I stay after when I have 9 patients and 2 DLTs. So you can see now I'm using all of this data to estimate the DLT probabilities at each dose level. Again, we're using a simple statistical model to do this at the end of the study only. We don't use the model throughout the study like we do in CRM. And this 2/9 is a 22% DLT rate observed at dose level 3. That's closest to my 25. So that would be my MTD recommendation at the conclusion of the study. This also has a web application with easy to use interface. one of my favorite parts is this trial protocol tab. You can actually generate a stat section for the information that you input into the app and use that in a protocol. And it's very, very convenient and efficient. So use of these novel designs require collaboration with a biostatistician. I was part of a team that wrote a study with 2 statisticians, including myself, and 2 clinicians, and just our experience and how we collaborate on using novel designs in phase 1 trials. You can check that paper out in JCO Precision Oncology, and it has a good starting point for conversations between clinicians and statisticians on how to design these trials. So I'm going to use the last few minutes to talk about more complex settings very briefly. So on the left side of this slide, we have a traditional setting that we've talked about already, and this is defined by dose levels that are ordered with respect to the probability of DLT. So we saw, and efficacy. So we saw as we increase the dose, we increase the probability of DLT, but also the probability of efficacy. So we're going to relax that assumption and talk about drug combination studies. On the left side, we have a single binary DLT endpoint driving the dose allocation and MTD recommendation. We're going to relax that and talk about multiple endpoints, maybe using efficacy and toxicity to make a dose decision. And then DLT observed in early cycles drove dose allocation and MTD recommendation. And we're going to talk about the case in which we have late onset toxicities and what we would do there. So here's an assumption about the DLT endpoint that is inherent in the designs that we've talked about so far. So let's say we have a 28-day cycle that we're using as a DLT evaluation window. And this patient, this may be a slightly unrealistic case, but it illustrates the point. If the first patient here starts at week 0 and goes for 4 weeks, 28 days, and it fully completes the DLT evaluation window of 1 cycle, and then the second patient comes in here on the orange bar, 6 weeks after the start of the trial, and then is observed for 4 weeks. And then we have their completely observed, fully observed DLT evaluation window information. And then the third patient comes on. This is the assumption being made in CRM and Boyne that we have these fully observed observations as we make dose assignments. But with longer DLT assessment windows or fast accrual, this could happen with either one of those or both of those situations. We might have a really long DLT assessment period here, perhaps of 4 cycles of treatment. And while we're waiting on the outcome for the blue patient, the orange patient comes on study. We have about 6 weeks worth of information for the blue patient when the orange patient comes on. And then we have about 6 weeks worth of information or 8 weeks worth of information on the orange patient when the green patient comes in. This is probably more realistic case that patients are coming in while we're waiting on outcomes for previous patients. So the question becomes, how do we use this missing or pending data to make dose allocations if we need to assign the orange patient a dose and we're still waiting on data from the blue patient? How do we make these assignments? So this is a setting called late onset toxicity. And so the question becomes, how can new participants receive timely treatment when the previous participants' outcomes are still pending? Do we assign all to the current dose when we have a new patient? That might be inefficient allocation. We might assign a lot of patients to the lower dose levels if we do this. Or do we wait until a cohort of 3 completely finishes the DLT evaluation window? If we do that, we're going to have patients waiting to be assigned. We're going to have long trial duration. So neither of these cases is optimal or very efficient. So the methods we just talked about, Boyne and CRM, they have extensions to handle late onset toxicities. These are called time to event methods or TIGHT methods. So there's a TIGHT CRM, there's a TIGHT Boyne. And what you do here is specify a longer DLT evaluation period to capture those late toxicities. And we use partially observed data based on the amount of the DLT evaluation window each participant has completed when a new participant is accrued. So for example, if the blue patient has completed 5 out of 8 weeks when the orange patient comes on study, that patient is basically weighted as 63% of a non-DLT outcome at that point. And so we use that partial information to make the dose assignments in both of these methods, tight CRM and tight BOIN. So it's basically a way to make the dose assignments when you only have partially observed DLT data. And there's software for these extensions, time to event BOIN at trialdesign.org. There's a tight CRM web app at this address that's located on the slide. And so again, these software applications are enabling facilitating more use of these novel designs because of the ease of use and the efficiency to be able to implement these types of designs. So let's talk about another challenge that we highlighted a few slides ago, and that is escalating more than one agent. So I'm talking about drug combination studies in which you have dose levels of agent A and dose levels of agent B. And the main challenge here is if we're at the green combo, which is dose level 1 of each agent, and that's deemed safe, I don't have a DLT there, where do I go next? Do I go up this column to the red one, or do I go across this row to the blue one? The challenge here is that we don't actually know. We have some uncertainty whether or not blue or red is more toxic. So a movement from blue to red requires an escalation of one agent and a de-escalation of the other agent along this diagonal here. So that creates some uncertainty as to which is more toxic. So we want to account for that uncertainty in the design and not assume that we know sort of the ordering of the dose levels here for every dot on the screen. So here's what's done in practice here. This is an old review, but in 2015, in Annals of Oncology, 162 drug combination studies were reviewed in which 2 agents were escalated, and 161 of 162 chose a path with some known ordering and applied some single agent method like CRM or BOIN or 3+3. So that can happen in multiple ways, right? If we just look at the blue dots on this left side, right, I know that each time I move to the next blue dot, I'm escalating the dose. There's no uncertainty because we're not accounting for anything along a diagonal here, right? So what that does is allow me to use 3+3 or BOIN or CRM and just explore the blue dots. But what I'm leaving out is the gray dots. My optimal dose combination might be the gray dots, and I'll never find it if I take this approach. Same with the right side. If we just look at escalating both agents at one time, we go up this middle diagonal, we're leaving out all of these gray dots and possibly leaving out a promising combination there. So BOIN and CRM have been extended to this setting as well, in which we have a BOIN combo method and a web app for that, and a partial order CRM or CRM for drug combinations that also has an app here available at this website. So lastly, I'm going to talk about, should we be targeting the MTD? Just a few more slides and we'll wrap up. This is a hot topic right now, and one that I hear a lot about: dose optimization. Project Optimus from the FDA. This is a figure or a slide from this Cancer Letter article in 2022. That the FDA and others are calling for the MTD era, and more is better, right? Pushing the dose higher, as we've talked about so far. They're proposing to end that era and try to dose optimize based on other endpoints besides safety only. So this is the focus of Project Optimus, which is reforming dose optimization and dose selection paradigm in oncology. So let's revisit our assumptions. On the left side is the original assumption we had for cytotoxic chemotherapy, in which we are increasing the dose. We're pushing the dose higher because that up to this threshold, because toxicity and efficacy increase as we increase the dose. And on the right side, this again comes from this Tracy Cancer Letter. Again, the assumption is that toxicity is increasing as we increase dose, but efficacy is sort of leveling off at some point. And a lower dose may be nearly as efficacious as a higher dose. And beyond this green arrow, we're just adding toxicity. We're not adding much efficacy. So we really should be targeting this more optimal dose based on toxicity and efficacy rather than pushing that dose as high as possible up to the maximum tolerated safety threshold. And just like the other problems that we've been discussing, complexities in early phase trial design, BOIN and CRM have adapted to this type of setting, this paradigm. And there's a BOIN 1-2. This is usually sort of phrased as a phase 1-2 trial in which you're using a safety and efficacy endpoint. And there's a BOIN 1-2 for efficacy and toxicity dose finding, a CRM extension to this as well, both with software to be able to implement these types of designs. There's some endpoint considerations that should be taken into account. What's the efficacy endpoint that will be used? Is it overall response? Is it some biomarker? That really needs to be part of the conversation. Most importantly, when is it scored relative to toxicity? Because if efficacy is scored much later, right, you're going to run into the pending data problem that we saw with toxicities earlier. If we get the toxicity endpoint in 1 or 2 cycles, but we have to wait 4 cycles for the efficacy endpoint, it's going to cause some issues with pending data, and we need to account for that in the design. So here's the 2 designs that I showed you earlier with BOIN and CRM extensions to phase 1-2 trials. Those are assuming that you have efficacy and toxicity data fairly observable in a fairly short period of time, and you're using both of those endpoints to make adaptive dose assignments. Another approach is to use a more sequential approach in which we do a dose finding study in which we select a range of doses. So maybe we do BOIN or CRM to select doses 2 through 4, get some safety data, select doses 2 through 4 to carry into a dose optimization sort of randomization phase. And so basically the dose escalation to dose optimization phase, the transition there is just to whittle down the doses from 4 to 2 and have some better idea of sort of what doses we should concentrate on. And then in this phase, you take more account of the efficacy and the toxicity, and as opposed to the dose escalation, you just take account of the safety. And so you're doing this randomized dose optimization. So this is another approach to this type of problem in which we're trying to optimize dose. And with that, you need some strategies to accumulate data at multiple dose levels. So I don't have time to talk about this today, but there's a lot of talk about backfill in oncology dose finding trials in which we might be currently at dose level 3, and we might put the next cohort there, but then we're going to also backfill to dose level 2 and get some additional information there. And closely related to that is randomization in phase 1 trials, in which we're randomizing to multiple dose levels, again, to try to obtain data more broadly. So this is my last slide. This is the concluding remarks. Novel statistical methods are being developed in early phase trials, but not used very often. And with the software that's available, especially with protocol statistical templates that are out there, the difficulty shouldn't be a barrier like it was 10 to 20 years ago. Don't let the single agent design structure limit goals of the study. So really, this is a general clinical trial comment. What research question are you trying to answer? And try to design a trial that answers that question, right? So sometimes I've had clinical investigators come to me and say, oh, we really want to escalate both of these agents, but we're not sure if we can do that. And so, we really talk about, okay, what question are you trying to answer? And now let's design a trial that answers that question. So really tailor the design to the science and the questions that you're trying to answer and research objectives you're studying. And as we saw towards the end, you don't have to target the MTD. You can optimize dose based on multiple endpoints in multiple different ways, using both of those endpoints simultaneously and making dose assignments that way, or a more sequential approach in which we get a small number of dose levels to take to a randomized setting in which we dose optimize in that phase 2 trial. So that's all I have. Questions are welcome, and you can email me at this email address with any other questions that you have after the session.
Thanks. Appreciate that. It was great. I got maybe 2 comments. I mean, I don't do a ton of sort of early phase stuff, but I think the first bullet point of your summary slide was, this shouldn't be so complex anymore, et cetera. I don't know. To me, it still is pretty complex, and you need a really good involved statistician to do this, which not every institution has. And so it's not a criticism. It's just that, I think if you're at those institutions or have those stats people, you can do this. Otherwise, I think a lot of institutions struggle to really do these completely appropriate but more complex designs. My other comment is maybe the slide before that had that. Yeah, we see a lot of this in industry trials now where they do some phase 1 stuff and then their sort of phase 2 lead into phase 3 is a few different doses, sort of based on Optimus, right? That's almost everything I see now is dose A, dose B plus drug X, whatever, whatever it is. But which I don't think is a bad thing. I get Project Optimus. I think it obviously introduces delays. So I don't know. We'll see how they don't really have a choice, by the way, but I'm not sure that that's necessary for every single trial or every single approach. And then my last comment, and this is more of a question, is, I did a lot of sort of TKI dose titration 10 years ago, got a lot of gray hairs based on it. And it occurred to me at the time that we're sort of trying to find an MTD or the average dose sort of across a broad population. But what we should really be doing is optimizing dose in an individual, right? Because a certain dose of especially oral drugs will produce different drug exposures for everybody on this call, right? And we have no way of knowing that. We don't really measure that, et cetera. And so rather than testing different dose levels, it should be testing different titration schemas to say, well, this is the schema that gets people to that optimal benefit risk. So I guess I'm just wondering your thoughts on that. I know it's probably super complex to do, but it always seemed to me like that was a missed opportunity for oral agents.
Yeah, I think, thank you for the comments and the questions. I think bridging the first comment you made with the last one and the question, right, is that I think we should be doing that. Can we account for patient heterogeneity in these doses, right? And can we account for interpatient dose escalation, right? If somebody start that particular patient at a low dose level, and if they're, depending on their outcome, titrate them up. I think that's what you're talking about there, right? In sort of some.
Yeah, not so much dependent on outcome, although that could be incorporated, but more just, a schema of titration that gets them to their own sort of optimized benefit risk.
But maybe not based on the outcome, but you are talking about escalating a dose within a patient. Yes. Yeah. Right. Yeah. So I think both of those things are topics that people have researched as far as statistical methods go, but I think we're far away from that due to the comment that you made before, right? Once you, the first comment you made, once you change the complexity of this, right, it becomes that you need a heavily involved statistician. That's what I do. I'm a heavily involved statistician right. with these types of trials, but I recognize that that's not the case everywhere, right? That's not always the case. So, I gave this talk somewhere. I gave this a similar talk. I gave a drug combination talk once, and somebody raised their hand and asked me if I'm on call. Do I have a phone and people do people call me with the updated data and I give them the dose recommendation? So it's not quite like that, but I do have to be available in a timely fashion to take the data, make the dose recommendation, and update it to a system and all of that stuff. So there's a lot of implementation challenges with this, right? Mm-hmm. I think once you implement or incorporate patient heterogeneity or interpatient dose escalation, the problem just becomes much more complex. And we need, I think this, in my view, this comes down to we need better support, better funding for clinical trials. I know we wish we all had that. And then that could support personnel that can do these things. If you really want to do it,
no, I agree.
We need mechanisms that adequately support these financially, I think, and that includes a biostatistician.
Agreed. Yeah. Rainier, there's a question.
Yeah, thanks. My question is how's the FDA perceives these kind of trials and whether there's pushback and they're part of the reason why some of these kind of methods are not as common.
I think Boyne actually is becoming much more popular, and well recognized and accepted by the FDA. I haven't gotten any pushback about using that. I haven't really gotten much pushback. I've used CRM several times, and my pushback from the FDA there has not been don't use this or this is problematic. It's been we want to see what happens if this, and they give me some hypothetical, like what if the first dose level is too toxic, or what if the last dose level, or none of the dose levels have high DLT, like what happens in these situations. And what we do is provide simulation results that give them an idea of how the design behaves in those particular scenarios. So the pushback has been, can you give us more information? But it hasn't been, don't do this. That's been my experience. But I think Boyne has been a little more accepted because the transparency there is much higher, right? You can give them a table of how the dose assignments would be made. Everybody can see it, and it's been well accepted. The last two trials I've designed that I've started with just a Boyne to find the MTD. The FDA said we want a more Project Optimus approach in which we find more than one dose to try to dose optimize in the next phase. And so I've used like a backfill approach that accumulates more data at lower dose levels. So the pushback there has been not don't use Boyne, but don't find the MTD. They actually want you to find multiple doses so that you can explore multiple in the next phase.
Thanks. Maybe last thing, I'm going to pick on our patient advocates, Tina, Salvo, Stephanie, I think, or maybe others. Like, how do patients view all this, right? I mean, in the traditional 3+3, is, I'm getting a low dose, probably not going to work. I wouldn't want to be that patient. Then intra-patient dose escalations come along, but I don't think commonly used. So I just sort of curious on your thoughts about the general approach to dose optimization from a patient standpoint.
Well, I can say, I mean, we run into problems with patients not wanting to enroll in phase 1 trials because they're worried about getting suboptimal doses. We just had this exact thing with a patient enrolling in a chromophobe trial, for a trial for non-clear cell. So this is really enlightening and helpful. So thank you for the presentation. I'm actually shocked that the FDA isn't pushing for these new designs, because to me it seems like this would be better for patients. So that would be my surprise, that there isn't regulatory efforts to make this happen. I don't know what you think, Salvo.
Yeah, I have another thought that I want to ask Nolan, and this Boyne designs and the other design that are kind of shifting away from the 3+3. I mean, from a patient perspective, would you agree that this is more like a shift towards being more cognizant about toxicity and those optimizations that goes towards the paradigm of more like towards the Optimus project that the FDA is pushing, rather than focusing, even though these are like, we're talking about designs, but the design that takes into consideration toxicity rather than a design that takes into consideration optimization of those. And that is, I think, a concept that patients would resonate with, knowing that they're enrolling in something that is optimized to be more like towards that optimal dose rather than hitting that MTD. I don't know what your thoughts are.
Yeah, I think I agree with that. I think, the quote optics of being in a trial that you're dose optimizing rather than, finding a MTD just on safety perhaps would be better. But I still think one problem is going to be largely those trials start the same way, right? You have to start low at a low dose level. You have to escalate up, and usually you get pushed back if you try to escalate in cohorts of one patient, right? So you can't move that fast through the dose levels. There's pushback against that, right? So largely these trials start the same way. And so if a patient's worried about being put on a sub-therapeutic dose level, that's going to remain early in the trial, right? It might be better later in the trial, But early in the trial, the first 6-9 patients, they're going to be put probably on suboptimal dose levels, subtherapeutic dose levels. And until we get to a place where we're like, and I think part of that is starting at a low dose level. If you're in a 3+3 mentality, the mentality has been you're not going to get to a spot and then go back down to dose levels that you've already tried, right? You're going to get to a point, and then once you see 2 out of 6 DLTs, you're just going to declare the dose level below that, the MTD. But Boyne and CRM will move through the dose levels to where if you see something too toxic, you can go down. So it really would lend itself to, let's start at a dose level that we think is the optimal dose. And if it looks too toxic, you go down, right? But we still are in a mentality of start really conservatively, in my view, start really conservatively at a really low dose level. And you're putting a lot of patients still on subtherapeutic dose levels if you do that. And I think there's a case to be made for that. We want to be safe. We want to be conservative in terms, but then the patients that are worried about being put on subtherapeutic dose levels, that's probably still going to happen no matter if you're dose optimizing or finding an MTD.
Yeah. Anybody else? We got a couple minutes left. I think it's really interesting.
Yeah, I'll just make one more comment if there's not another question.
Sure.
Right off the bat. There's one thing I didn't get to in this talk, and I'd be interested in anybody's thoughts, especially the patient advocates. There's a growing interest in using PROs in this phase of development, and using a PRO CTCAE to account for patient-reported adverse events, and what that entails, like patients, how they report things, because they might be afraid that they're going to be taken off treatment if they accurately report their AEs. And there's just a lot of talk about that right now, and I think that's a very interesting topic as well. How do you use a clinician-reported and a patient-reported adverse event to make a dose decision? So all this comes down to, how do we use multiple pieces of data to make dosing decisions and try to optimize that dose? But PROs, I think, is a place that you're going to see more and more attention soon. So
I mean, I think the current PROs that we have, especially in RCC, are really suboptimal and probably not representative of what matters to patients. So that's my biggest concern with the move in that direction, until we have better data that's more, I think, reflective of the patient experience. We do a lot of surveys with patients, and interestingly, patients are not worried about toxicity. They're worried about efficacy always. You know, that is their driving thing. And I think there's sometimes such an effort to avoid and keep them from getting toxicity as well. There should be, but at the end of the day, what patients want is something that will fight against their cancer. So
yeah. Thank you.
Well, thanks, Noah. We really appreciate this. It's been insightful and I think useful for everyone. And thanks to everyone for joining. So